Nobody asked me, but I have the microphone, so you have to listen to me. So what are we going to talk about in 20 years in Mifobio? And if you ask me, I would say we're going to talk a lot about actually computation, artificial intelligence, and analysis. So, and that is actually some of the things I want to show you today. And I'm kind of a little bit afraid that at the end you'll get away with the idea that we're not going to do FCS anymore, actually. We're just going to do actually CNNs, deep learning or so. But let's see. Okay. So before I start, let me thank very much for the people who worked on this, Waiho Tang and Xiao Ren Sim. They have developed all the CNNs. Daniel has developed part of the computation. He's done actually all the development of the instrumentation. And Ashwin helped us with some of the superficial measurements. And Adrian is actually a statistician who helped us actually develop some of the CNNs. Okay, so fluorescence correlation spectros. Give me a short introduction because Don Lamb told me I have to do this because I took his slot. So if you look back at 1967, Manfred Eigen received the Nobel Prize, and he got the prize for the studies of extremely fast chemical reactions affected by disturbing the equilibrium by means of very short pulses of energy. So what that means is actually he had actually a system. There's one parameter he observed over time. Then he created a jump and a variation of that parameter. And then he looked how that system came back into equilibrium. So he disturbs the system, looks how it goes back to equilibrium. From there can actually deduce an actually information about the system. Then there were then in the 70s a couple of people, that was Douglas Magde, Elliot Elson, and Ward Webb in the United States, and that Rudolf Riegler and Måns Ehrenberg in Sweden, who realized that, well, if you think about this, right, if you look at the signal, you constantly have these fluctuations. These fluctuations are nothing else but actually this perturbation from the equilibrium and spontaneous relaxation, constantly. So actually, that means if you use actually these fluctuations, you should be able to get the same information out. And you can measure an equilibrium. We just look at the fluctuations. And that's what they did, and then they used actually some things called autocorrelation functions to analyze these fluctuations, get actually information about diffusion coefficients, chemical reaction rates, etc., etc. So this autocorrelation function, it looks actually like this. It means actually you take the average of a signal, f of t, the fluorescence signal, compare it with the signal a little bit later to compare whether the signal somehow persists in time. How does it compare to the current time, right? So how long does any kind of fluctuation persist? And then what you do is you take these two signals basically, and that's what you see here on the right side. You see the blue one is the original signal, the yellow one is the one shifted in time, and then but for every single shift in time, you compare how well they overlap, and that gives you your correlation function. So you see actually at 0 time, when there's perfect overlap, the correlation function is maximum, and then if they have more and more shifts, the correlation function decays, and it decays in the way that when the fluctuations slowly de-align, right, then your correlation function basically has dropped. That means actually the width of the correlation function tells you something about the width of these fluctuations. And that allows you then to characterize these fluctuations, where they come from. So how do we use this in FCS? So in FCS, the original versions, or let's say the common version, is a confocal FCS system, where you use a confocal volume. And if you have now a small molecule that goes through that focal volume, it will create a fluorescence peak, right? It will create exactly what we just talked about, these fluctuations. And if you have a larger molecule here, if it goes through the same focal volume, it will create a much longer correlation, right? It takes a longer time to go through that focal volume. So now what you have to do is, if you do this very, very often, many transitions, you can calculate the average time a molecule needs to go through that focal volume. And from there you can determine how a molecule moves. That can be diffusion coefficient, that can be transport and flow, etc. Right? And this is encoded in the correlation function, as I just showed you on the last slide, by the width of the correlation function. So the wider the correlation function, the slower the particle moves. Right? So we can recover things like diffusion coefficients. The other thing you can recover from it, at the moment here I was assuming that you have actually always one molecule going through. Well, sometimes you have higher concentrations, so you might have multiple molecules here already, right? So if that's the case, then you get already a relatively high intensity before, but if a new particle comes in, again, you get your fluctuations, right? So what that means, you have actually two different levels of intensities. The first one is the average intensity. That depends on the average number of molecules you have in your focal volume. The second one are these peaks, right? They come from single molecules, right? So you can imagine from there already, because you have these two informations, you can actually deduce somehow as well the concentrations in the focal volume, right? And that's what is encoded in the correlation function, the amplitude, where the higher amplitude means lower concentration, lower amplitudes means higher concentration. Yeah? And for those of you who, I mean, just to give you an idea why, here I give the example of the restaurant upstairs, right? When you go there later at 12:30, it's going to be completely packed. If one more person comes in, you will not realize that, right? But if you go there now, I hope it's empty, because you're all here, right? So if one person comes in, you would immediately see that. So if you have lower concentrations, fluctuations are easier to see. And that's exactly what the correlation function tells you. If you have low number of particles, the fluctuations are easy to see, you get high amplitude. Okay, so, and since the 80s or so, we have used actually really this confocal FCS in many instances for very interesting measurements. But there's of course one problem with that: you get only 1 point. So if you go now and want to, for example, get the diffusion coefficient on a cell, or every single point on a cell, you would have to scan that cell and every point measure the correlation function. A correlation function, we need typically at least 10 seconds to get the full data. Which means if you go over a cell which has perhaps, I don't know, 20 times 20, 30 times 30 points, you're talking about hours of measurement. Now, if you measure, I mean, I don't know what you do typically with confocal microscopy, but if you measure a cell with a confocal microscope continuously for 1 hour, I don't think that cell survives. And if it survives, it is not the same when you start at the left upper corner and you come down to the right lower corner, right? So, so we need a method to do this in parallel, and that is then what we call imaging fluorescence correlation spectroscopy, where you use a light sheet or any kind of illumination that allows you to illuminate a cross-section of your sample. And then you, you project the fluorescence from that light sheet that's created by light sheet onto a camera. And what you see here, if a particle comes in here now, right, it's projected onto the camera, and when it goes through a pixel, right, to the first pixel, when it goes through, you'll get a peak, or it goes to another pixel, you get a peak, right? So now at every single pixel, you get basically your confocal volume. Now at every single pixel, I can actually calculate my correlation functions. And what that looks like then, well, I should perhaps start. So what it looks like then, for example, they get actually a large amount of correlation functions, right? The blue ones are the data, the actually real data. The red ones are the fits to these correlation functions, right? And then you can extract the number of particles and the diffusion coefficients and provide them in maps. That's what you see here, right? And here you see the projection of the sample. In this case, it was a Drosophila sample where bicoid was labeled with GFP. Right? So when you see here now in the nuclei and around the nuclei, the concentration and the diffusion coefficient. Okay, so now that's actually really nice. So you can actually get really a lot of measurement at the same time. In our lab at the moment, there are two different ways we do that. Either we use a total internal reflection microscope, because in that case, you really illuminate only the first 100, 200 nanometers of your sample. It's ideal for cell membranes. When you want to look at that, and we work a lot on cell membranes and cell receptors, right? It has very high signal-to-noise ratio, as you know, because in turf you don't get any propagation of the light on the sample. So that is a very good system. What you can do in turf, you can do, some people sometimes call dirty turf or variable angle illumination, right? You go out of the critical angle, and then you kind of create an inclined light sheet in your sample. You can do that as well, right? As long as you get some sectioning in the Z direction, you can do the imaging FCS. And that's what we use as well in the last couple of days in the workshops here. Or of course you can use actually a light sheet. In a light sheet, what you do is you have an illumination objective that creates your light sheet, and then under 90 degrees with a high numerical aperture detection objective, you project the fluorescence from your sample then onto the camera, and again you can do your imaging FCS. So I'll just give you a couple of examples what you can do with it before we then start actually really looking at AI. So that is an example of a zebrafish. For example, we were interested in the cerebellum, in the brain, of a particular molecule called Win3. So you can focus into zebrafish brain. You focus on one cell and go to the top cell membrane, and then you can record your correlation functions in the fish. And here this Win3 EGFP, and here we have Win3 EGFP under drug treatment, and then you see that you can actually record the difference in diffusion coefficient. In this case, the drug treatment removes actually some of the lipidation of the Win3, so it starts moving faster in the membrane. So it's actually and that was done with a light sheet microscope in zebrafish. So you can do these measurements not only on biolayers or cells, but you can do it as well in animals. Another thing we've done last years is, now, since you have actually all pixels at the same time, you can ask the question, okay, how long does it take actually for a molecule to go from this pixel to this pixel? That would be called a spatial cross-correlation function, right? So we are trying to figure out, like, this molecule is like, how long does it take to go through this other pixel? And you can do that. And what you would get then as a correlation function looks a little bit different. You would get something ideally that has a nice peak, where the peak position tells you how long the molecule needed to go from point A to point B. In principle, it's even a better way to measure your diffusion coefficients. But with this one, you can measure directionality. So again, we have done that in a zebrafish. That is another morphogen, a molecule called squint GFP. It was expressed in a very early fish, 5 hours post-fertilization. And you see there these channels. And what we were interested in is how these molecules move in these channels depending on the channel sizes. And then you can see the correlation functions. And for those of you who have been in the workshop, I hope so you realize that they look a little bit better for the cross-correlation, for the spatial cross-correlation. And then we can actually measure the diffusion coefficient in all these different channels and determine how the channel size influences the diffusion coefficient of that particular molecule over different distances. And then, of course, the last thing which we have tried, I think, over the years is actually fluorescence cross-correlation spectroscopy, just to give you an idea of that. So you take two different colors, and if you have two molecules that move at differently, right, they're not interacting, then of course what you get is actually you get these fluorescence peaks, but they're not correlated, right? Because these two particles move independently of each other. So what you will get, you'll get your autocorrelation functions, right, the red and the green, that will tell you how fast the red and the green particle moves and how many they are. But the blue one, the cross correlation, will be zero. It's like none of them are interacting. They don't move together. If by contrast you have something that moves together, like here, then these fluctuations always happen at the same time. Now, if you measure that often enough, you'll get ultimately a cross correlation function, which tells you now how many of these particles actually interact, right, and how they move together. And you can do that as well with imaging FCS. So what we show here, that's different cells. In one, we have a negative control. That is two membrane labels that are not interacting. Then you get actually, here dark values means very low cross-correlation. Then here we have an EGFR receptor that contains two labels, red and green. That means they always have red and green together. Right? So then you get that your positive control, you always get very high values. And in this case, we have two EGFR, one with green, one with red. And then we start to like, do they actually interact? And interestingly, in this case, we see that yes, they do interact. And for whatever reason, they seem to interact more strongly on the periphery of the cell and less strongly in the center. So these are the kind of things you get. And this one is, I think, something we're still working on, because in an image like this, that means I can give you an image of the KD of interactions in a whole cell at every single point, right? That's what I really like about that, and so we are still working on that to make this actually more accessible. Okay, so what's new? So what I want to talk about here in the rest of the time I have. So the first thing is we have developed, that you have seen that in the workshop already now, the software that you can get the correlation functions you measure, calculate directly and shown while you do your measurements. And I show you how we can use that. Then I'll show you a little bit on how we can accelerate our evaluations with GPUs so that we ultimately get actually real-time measurements. So you do your measurements, at the end of the measurements you know what you did, right? And then I will talk about how we use CNNs, so deep learning, to accelerate actually this even further. And then what I'm not going to talk about, but that's for me, that's I think the future. The future for me is that we're going to use multiple CNNs together with GPUs, right? And we create a fully automated workflow for your measurements. You basically go there, put your sample on your microscope, tell the microscope to align itself and do the measurement, and you come back after a coffee and a croissant, and then you have your measurements. That's the ideal thing. And you never have to actually fit anything, because people in general seem to dislike fitting data. Okay, so very quickly the power of this correlation spectroscopy approach is if you think about a SPIM microscope, right? You have two objectives and they have to be aligned to each other. So there are two things. First of all, you want to have that the light sheet that is created by the illumination objective is actually in the focal plane of the detection objective. That is one thing. The second thing you want to have is that the thinnest part of the light sheet is right in the middle of your field of view, right? So you basically have to align these two microscope objectives, right? And how do you do this quantitatively? And one way to do that is really simply to look at the correlation functions. You put something like a beads or flow force into your sample in a liquid, and then you measure correlation function. You see if you, for example, defocus your system, right, or change the focus of your system, your diffusion coefficient starts increasing until it hits a maximum and decreases again, and the number of particles decreases until it hits a minimum and decreases again. And of course, the fastest diffusion and the smallest number of particles happen at the same time. That is when you're actually in focus. Because you can do that automatically, while if you have actually a piezo or something to move your objectives, you can actually basically tell the computer, go ahead, measure, let me know when you found the right position. And here on the right side, you see the correlation functions. So here you see that the black correlation function has a highest amplitude, lowest number of particles. And here we normalize the correlation function, so you see that the black correlation function has as well the narrowest width, right? So it has the largest diffusion coefficient. And of course, you can align as well a dual view, right? If you split your light on the camera into two wavelengths, red and green, you want to do the cross correlation spectroscopy. Now, you can do exactly the same thing. You can take a look at the cross correlation functions, right? And you can start aligning them. Just take a look at the central panel here. You can start aligning these pixels until they fully overlap. And you see how the amplitudes of the cross correlation function go up until they reach a maximum. So this way allows you as well to align your even a dual view. I don't know so perfectly, but better than people can do it by eye typically. Okay, so we can do all these things. So why do we need deep learning, right? So, and I'll try to give you a couple of reasons for that. So there was a really nice paper by Michael Culbertson et al. In 2007, Analytical Chemistry. And what they did is instead of fitting their data with a simple model, they measured the actual point spread function and compare the actual point spread function they measured to the theoretical ones we use for fitting, right? And said, okay, what I'm going to do now is I'm going to simulate my correlation functions until I get an overlap with my data. That, of course, took them 8 hours for one correlation function or so, right? But it is really the problem of reality and theory, right? And we always pretend that in FCS we fit with an ideal model, but we don't. It's an approximation, and on top of it, it's an approximation even for the ideal situation. But on top of that, your ideal situation you typically never reach, right? So you, in principle, could get better data if you do that. So Yeah, that's what I actually just told you. I should have clicked through that. I'm sorry. And with the CNN, right, with deep learning, we can do this now. We can actually take any kind of situation we want. We simulate the data. We train the network, and then the network can then actually predict the values on our in our system. And you can align this to your own system, right? So for your idiosyncrasies of your own system, whether whatever the PSF looks like. So this is really powerful because you become now agnostic of any fitting model. You don't care about that anymore. So the second problem is what I call the crosstalk and the problem of analyticity, and that happens in 3D. So if you think about a spin measurement, right, and if I take pixels that are of confocal measurements, you know that already. I have to place confocal measurements far enough apart if I went to measure them at the same time, because otherwise they would crosstalk. So that's why the spinning disk microscope you use has actually pinholes that are actually a certain distance from each other. If you make them closer, you get too much crosstalk and you can't measure anymore. Okay? So, and for turf we don't have so much of a problem because our layer is very, very thin, about only 100 to 200 nanometers. But if you look at the wide field or if you look at a SPIM, now neighboring pixels have actually a relatively large crosstalk, because actually different areas out of the focus will crosstalk to the neighboring pixels. And this leads now to a real problem. Now we cannot calculate anymore real analytical models. There is no easy solution. So what we've went through is we take numerical models. But if you ever have fitted with a numerical model, it takes very, very long. And that is something, again, you can get rid of with CNNs, as I'll show you in a moment. Then extrinsic versus intrinsic time resolution. FCS, I told you, takes at least 10 seconds. Actually, in reality, many people will measure 30 seconds or longer. All right? So, and that limits you. Because what that means is that if you have a system that changes only very slowly, okay, no problem, right? So if you measure actually a certain, let's say 10 to 20 seconds or so, right, that's fine, right? You get an average value and you're fine with that. But if you have a system that varies relatively fast, then you don't capture actually the changes in your system anymore, right? You ideally want to be faster than that. You don't want to be at 10, 20, 30 seconds. You want to be ideally at a second or 2 seconds or something like that. Right? So ideally, we would like to use less data for our evaluation. And I'll show you that CNN can deliver that for you as well. And a problem that another problem is if you take actually not enough data, right, your correlation functions tend to be biased. So if I take only 2 seconds of data for a correlation function, it will give you a correlation function. You can fit it, but it will get a diffusion coefficient that is actually somewhat too fast, because you don't have enough statistics. If you then actually take more and more frames or more and more data points, you slowly converge to the actual value it should have been, which is 1 micrometer squared per second in this case. It's just a simulation. And that has been known in FCS for a long time. There's recently, Jochen Müller from Minnesota has provided some solutions to that, right? But I'll show you that actually CNN can go and solve some of these issues as well. Okay, so what kind of systems did we develop? So we have actually, if you think about the normal way we do FCS, we take about 50,000 frames, so about 50 seconds of data, all right? Then we correlate them, get the correlation functions, and then we take actually a model, then we fit our correlation functions, and ultimately we create our diffusion coefficient maps. That is a normal workflow. And what we've done now, we've created 2 different CNNs. The first one takes as input the correlation function. And then it goes through a typical ResNet kind of architecture and then estimates the diffusion coefficient map from the correlation functions. The other one, and this one of course takes 50,000 frames, right? Exactly like the fit. The other one, which we call IMFCSNet at the moment, has a similar structure, but this one takes the raw data. But it takes only 2,500 frames, so only 2.5 seconds of data, of the raw data, and then does exactly the same thing and predicts actually then your correlation functions, but I have to say this takes a 3 cross 3 pixel area. So it takes a little bit larger area, but only 2,500 points. Okay, so all the CNNs we have are trained on, sorry. All the CNNs we have are trained on simulations, and we simulate them because we work on membranes on something like 0.1 to 15 micrometers squared per second. We train them on different concentrations, right, number of particles we see, different brightness of the molecules. We train them on 50,000 or 2,500 frames, depending. The time resolution we use is 1 millisecond. That's the NA in magnification for our system, emission wavelengths. And then the noise, I'll come to in a second. And then we train them on slightly different values of the PSF of your system. Okay. So in the beginning, we actually simulated 10 million correlation functions and started. Ah, thanks a lot. So. We use 10 million correlation functions and to train the CNNs. Now we use a little bit less than that. You don't necessarily need that much. But if you're more interested in that, we can talk about that later. Okay, so what we have to include in the training is a realistic noise model, because noise is the one thing you have to get right. If you don't get the noise right, then your CNNs very often typically don't predict actually what you think you see. So what we did, we took war stacks of an EMCCD, that's just part of it. You see the EMCCDs very often have patterned noise. Then we correct that patterned noise by something called the Hirsch correction. That was actually a +1 article in 2013, showed how to get rid of the patterned noise. And then now we get actually statistics of the noise of our camera. So these raw image stacks are with shutter closed. We just read out the CCD just to see how the noise looks like. And then we get a prediction, we get a distribution of the noise, and then we use that noise in our simulations to train the CNNs. In addition to the noise, to the EMCD noise, we add typically as well a little bit of extra Gaussian noise to account for other effects in your measurements. Okay, so let's start to take a look how these systems actually work. So what you see here, that's a comparison against simulations only at the moment. That's from FCSNet. That means it's the one that looks at the correlation functions at 50,000 frames. So here you have in logarithmic scale the predictions of the diffusion coefficient for a normal fit, a non-linearly square fit. And on this side you have the predictions of FCSNet. And we looked at 0.1, 1, 5, and 10 micrometers squared per second. And what you see is actually that overall the predictions are actually very well. The work actually very well. And at the 10 micrometers squared per second is actually the NLS that's actually a little bit off, because you start getting too fast for the low time resolution of 1 millisecond to measure that. We can do that in 3D as well, and you see the same thing, right? So we are pretty much all right on spot with the CNN. Right? So that works actually very well. Now it's like, I mean, of course it has to work on simulations, otherwise you're already dead in the water, right? So the comparison against simulations for the IMFCSNet with only 2,500 frames, that is in principle the same thing. So we show here orange and so the orange points are Gaussian, contain Gaussian noise in the simulations for training. The black ones here called EMCD noise, and the open circles, these ones here, are the fits by NLS. And what you see now, so the ground truth we always take from the 50,000 frames NLS fit. But if you do this at 2,500, you see that the NLS is completely off, right? With 2,500 frames, you can't create correlation functions that fit the data, right? If you look at IMFCSNet, it lies right on the diagonal, perfect. And here in 3D, it's even worse. You see actually, you see it here already, but here as well, actually most of the data from the NLS, from so normal fitting, lies somewhere up here. Out of, it's, yeah, okay. So, so that shows you that these work much better. Now, if you compare this to a lipid bilayer. So here again we have the CNNs versus a fit. And then here you see actually the predictions. So the orange one is actually 2,500. You have actually now very good time resolution, right? And then the other two are the average for 50,000 frames. Of course, they give much less points, but you see that they, in the next slide, that they follow each other actually quite exactly. So what I've done here, we have done a sliding window approach. So we take 50,000 frames every 10,000 frames to show that the IMFCSnet, right, basically follows pretty much exactly what the fit actually tells you as well. So the CNNs actually work very, very, very well. And the last thing I really want to show you very quickly, one thing that happened to us, at some point we make a measurement and then the NLS and the FCSnet, they started actually predicting lower diffusion coefficients. And the other CNNet constantly predicted the same diffusion coefficient. I was like, we wondered what happened. So we took a look, and then actually what happens, we defocused. So, because of defocusing, the correlation function gets wider. And the FCSnet and the NLS can't make the difference. They're like, okay, you get slower. But the other one, the IMFCSnet that is trained on a little area, can make the difference because it sees how the patterns of the photons changes on this little area and automatically corrects it. And that was pure serendipity. That was not planned. Yeah, that is so. The CNN is actually robust against defocusing. It doesn't even matter anymore if you're actually in focus. And if you think about this a little bit further, in principle, that means you don't need adaptive optics in an animal anymore. Because if you actually train on a little bit larger area, you can actually train the CNN to get the correct diffusion coefficient out, even under cases where you have actually distortions of your PSF. So let me show you very, this one, that's the last one, okay? So again, the Drosophila embryo. So here you see actually a fit of a Drosophila embryo again. Here you see as the FCSnet, the one that takes the correlation functions. And here you see the prediction. You see the predictions like pretty much on the diagonal again. It looks like the FCSnet is a little bit biased. On average, it lies a little bit below the fit values, which we'd expect. But you see as well that the ellipse is a little bit tilted. What means actually is that the CNN gives you smaller errors than the actual fits. And the second one, you see here below, that's actually the one with 2,500 frames. And what you see here is this completely fails. So that's a classical example, and that's why I want to warn you as well. CNNs are not just good. Here, you make a prediction, it still looks like, you can kind of get the structure, but the diffusion coefficients are all off, right? And in that case, the reason for that is, you can see over here, right? The correlation functions we get, for example, on bilayers and so on, are beautiful. That's easy to predict for a CNN. If you go onto an embryo, right, they look actually much, much worse. They're actually quite bad. So what happens now, the fit as well as the FCSnet, they predict just from the correlation function what the diffusion coefficient is. The correlation function filters already more or less the problems. But in reality, we have actually a faster component probably in the beginning, and the IMFCSnet can't handle that because it sees only the raw data. It doesn't see the correlation function. So it gets thrown off completely. It has never seen the situation. It gives you actually wrong data. Okay, so last thing, GPU processing really helps you. If you 3D for the Drosophila, you take typically 28 hours if you try to fit this with a computer. If you use an IMFCSnet you get down to 41 seconds, right? If you use a GPU, you are still at about 800 seconds for the fit. You're about 6 seconds for the CNN, because the CNN, it doesn't have to fit. It's just it's run through the GPU and that's it. So it gets really fast and actually does all the things you want. So imaging FCS, I think can be automated, including alignment. We have real-time data treatment. Deep learning has multiple advantages, but you have to watch out. You have to retrain your system if you change your samples or your microscope. You really have to watch out for that. And you can use multiple CNNs at the same time for error correction. So if I take both CNNs, I will immediately see that they predict different values, so I have to take a look. That's one thing that one could think about. And we hope that the next step is really full automation of data evaluation. I just need some people who are crazy enough who want to do that. And for that, I'd like to thank, of course, actually, my group who did this. As you see, that is a little bit old photo. It was still COVID times, just to remember you of the good old times. And if anybody of you is interested, we have at the moment a special issue in Biophysical Journal on developmental biophysics. And the deadline, well, it's a little bit flexible. It's probably until, we extend this a little bit until March. So if anybody here is interested, contributing to that, please contact me. Okay, thank you very much.
Thank you, Thorsten, for the nice talk. If I got it correctly, when you train your neural networks with the simulations, it seems to me that the one using 2500 data set is even more robust in the 2D than the other one. Is it right? Is it also due to the fact that it uses an area?
Yeah. So if you think about it, I said 2,500, right? But it says 9 times 9, so it still has about 22,500 points. But it is actually relatively robust, and because you use the area, right? This actually can correct for other things like PSF change and stuff like that. So yeah, and I think it's well, it's more robust for the 2D case, but you saw for the 3D case.
No, for the 3D it was super clear, but
yeah.
Okay, thank you.
Thank you for the nice introduction for tomorrow. So I was surprised a little bit that the correlation, if you measure too fast, always gets shorter. Is that just because the faster correlations, you get more of them in this time period, that they don't average out? It just seems weird that it always shifts in one direction. I can understand if you measure for a short period of time, you don't get the full span of the fluctuations, but I would then expect it to get off randomly instead of always going to shorter correlations. But what you show is it goes to shorter. So
yeah, the correlation functions typically, if you measure shorter and shorter, the correlation functions start actually narrowing. So that's a bias you get in the correlation function. I mean that's what Jochen actually corrected for, right? But correlation functions are biased to shorter diffusion coefficients. If you
have more short fluctuations than long ones in that time period. Okay.
Hello, thank you for the nice talk. I have 2 short questions. One for the binding kinetics. So when you do imaging FCCS, I guess you're limited to rather slower processes because, like, what would be the fastest you can measure, like off rates of some 50 per second, or?
So okay, so we can measure. So typically we measure about 1 millisecond per frame, 1,000 frames per second. That's on the EMCCDs. If you go to scientific CMOS cameras, we can comfortably go down to 0.1 milliseconds. The fastest we've ever done, I see Don already smiling, it was 1 million frames per second. But that you can't, at the moment, that's crazy, right? I mean, we did this once just to show that it works, and then we never touched it again because it's way too much data. I mean, you can't really deal with it anymore.
For fast binding kinetics, you would still rather use stationary, conventional correlation spectroscopy.
Yeah. If you have to really go faster, I mean, confocal FCS is still the gold standard, right? I mean, you can go down to nanoseconds, right? I agree. We'll have to see. The SPAT arrays, you might actually solve a lot of issues. You can talk to Ignacio as well. He has actually a very interesting camera that reads out all the events. That might actually go faster as well.
Yeah, and actually the second question was related to the camera type. So you just said you use a back illuminated EMCCD or CMOS?
So in principle, you want to have a camera that has a high QE. Most of the cameras nowadays, QEs are 80% and above, and that is good enough. Otherwise, the camera type doesn't matter so much. It has to be able to to keep a stable temperature and to read out fast enough. And have a high QE. That's it.
Okay.
Almost everybody has this nowadays.
Well, maybe you can still tell me which exact camera you have.
So we have the photometrics 95B. You can use the Sonar from Endor. We have used the DU860 from Endor, the DU888, the DU897.
Okay, it's enough.
Okay.
We also have one of that type, so it's nice. We can also do something.
Any of them really works.
Thank you.
About that, the inhomogeneity of CMOS cameras, is it a problem or not?
Yeah, no, of course, yeah, okay, yes. So in principle, it is a problem because you want, if you get different noise on different parts, like you saw as well for the EMCCDs, you have to correct this for in some way because otherwise your amplitude is off. It's not so much the diffusion coefficient, but it's the concentration you want to determine. But the concentration is still something that's not fully solved on these cameras.
More questions?
Oh, super cool stuff, Thorsten. It's a poorly formulated question, but it was really nice when you showed this that your neural network could correct for the defocusing. And I wonder, eventually, I didn't catch, maybe you said it, but is it that, like, why is it that it's able to do that? Is it because it uses information from surrounding pixels, or like, what is the?
That's exactly it. Andrea told me to stop, so that's why I didn't explain this anymore. No, but that's exactly it. So if you look here, right, if you have a single pixel, right, and you transition through here, right, or through here, right, what you see that if your PSF changes, so you're out of focus, you get a different transition time, you get different diffusion coefficient, and there's no way you can see that on a single pixel. If you have actually 3 × 3 pixels, you have two informations. First, you have, of course, you have this information that the transition time changes, but you have as well the information that the pattern of the photons on the 3 × 3 array changes. So, and as said, it wasn't planned, but somehow the CNN must have figured out that, oh, you have a change in PSF, so your diffusion coefficient is actually that and not that. This is actually what bothers me with CNNs. I mean, they do something, we have no idea what they do. Right.
I don't know why. It doesn't work. I don't know.
Yeah, sorry, to follow up on this question about the spatial resolution that you may get from the image FCS. Can you give more detail about the training data that you give to the network? Because I suppose that if you have homogeneous or inhomogeneous training data, that may change the performance in the imaging.
Okay, so the training, we really simulate diffusion in a 2D or 3D situation, and we change all parameters, diffusion coefficients, brightness, number of particles, etc., and including as well a little bit the PSF. We give it a speed plus minus 10%. And then we train the network with that data. And you're absolutely right. So we train, for example, from 0.1 to 15 micrometers squared per second. But you can't predict at 15. You can predict only up to 10, because then your CNN starts actually tailing off. So we train an area wide enough so that we at the core we get actually good predictions.
And especially, like, you have differences in, you can train with different diffusion coefficient in different parts of the image, or is it uniform image?
That doesn't matter, because we actually we make the predictions by pixel anyway. So if different pixels have different diffusion coefficients, that's correctly predicted. Yeah. But the pixel size we have in FCS is typically only about the size of the PSF. So we are far away from super resolution or anything like that, just to get enough signal as well. Okay? Yeah.
Just a remark, if you train with 3D, maybe that's why you can deal with that, because it looks like a molecule that went out of focus during the training of the 3D. And so
exactly, it's the same thing. We get it in 2D when you defocus, and in 3D is the same thing. If you get a defocus, we find exactly the same dependency.